March. Statement. My name is Matthijs Welkers, as said, working at two locations. So one of them is the Amsterdam UMC, that's the Academic Medical Center in Amsterdam, partially doing clinical virology. And the second laboratory I work is for the Public Health Institute of the city of Amsterdam, at the laboratory working there. Now, for those of you not aware about Amsterdam, it's of course a city and a municipality in the Netherlands. It is really one of the main cities, and there's a big airport close by, and we have a lot of international travelers coming to Amsterdam. The total area of the city is about 200 square kilometers, and we have about 900,000 inhabitants, and a lot of tourism during the entire year, bringing and importing various infectious diseases that we monitor. For the monitoring, we have a special consortium set up, which is a combination of public health service in the infectious disease department that mainly does all of the source and contact tracing and epidemiological insights, and combine that with the laboratory work that's done at the public health service. And in collaboration with the Amsterdam UMC, the Department of Medical Microbiology, and also the Laboratory of Applied Evolutionary Biology, we do a lot of the phylogenetic analysis with the sequences that we obtain in the lab as well. And that combination, we try to translate as much to source and contact tracing and fighting an outbreak as much as possible. So that's a very good collaboration, at least, that we have. Now, concerning monkeypox in Amsterdam, we noticed, of course, the first cases in England coming up, and the messages coming from England, particularly one from the 18th of May. When we saw that message, we sort of sent out a word among all of the STD doctors in Amsterdam, be on the lookout, because maybe there might be a monkeypox case coming to Amsterdam. And it was already on the day after that people reported saying, hey, we might have seen a patient actually about a week and a half ago that could have been monkeypox. So we retrospectively tested this patient, which turned out to be positive and was our first case in Amsterdam. That was dated, we detected it on the 20th of May. Which was a visit from the 10th of May. And afterwards we had here you see sort of the epi curve that we had within Amsterdam and in the Netherlands. So our main peak, in total we had about 1,200 cases in total, and now on occasion we still find some monkeypox cases, but the large outbreak so far has subsided. We also were quite early, so this is the line indicates the moment that was declared a fact by the WHO, which was July 23. And at that time, we were already sort of over the peak in the Netherlands of the max number of cases that we had, already on a downslope. We're also one of the countries that actively vaccinated people, but at the time, at July 23rd, we were not yet vaccinated. We started about July 25th with vaccinations. We initiated it first in Amsterdam. So vaccination contributed, but it was not sort of the tipping point after which the outbreak started going down in the city. Now, after the declaration, of course, many other countries still increased in cases, particularly the United States. So we were first and already going down, and other countries were at that time coming up. Of course, immediately we wanted to do sequencing to see, what does it mean? How can we track infections? Can we detect outbreaks, whether or not they're linked? And I think everybody around the world was doing the same thing, using metagenomics. We also tried to do the same thing, extracting DNA using rapid barcoding, and then just run it on a single flow cell for 48 hours. And this is one of our first patients that or patient samples that we tried it on. You can see sort of the coverage that we got on an exponential curve. So it was about 10 to 20 times coverage over the genome. We found at least a lot of mutations in it, so it was not a very clear signal. In the end, we weren't able to publish it because the patient did not give consent to publish the sequence internationally, so we were not able to do anything with this sequence. but it immediately became clear to us because of full flow cell, it was very expensive and it was not scalable to the number of infections that we were having at that time. We had 15, 20 infections a day. So this was absolutely not feasible for outbreaks. So we sort of had to think of something else. And at the same time that this played, we also were trying to optimize our sequencing workflow for SARS-CoV-2. And we were doing that with a company that has sort of a custom PCR machine, which its design really goes back to the old ages of PCR with 3 hot water baths. They just have 3 different heat blocks and transfer the plate from heat block to heat block, thereby reducing sort of the PCR time from like 3.5 hours for the midnight protocol that we use from Nikki Fried to about 25 minutes. And when we were working on this, we immediately thought, like, yeah, why wouldn't this work for monkeypox? Why not? Let's just try it. So what we basically did was copied sort of the approach that was done for the midnight protocol, and in this way we increased the size slightly for the amplicons, so about 2.5 kb, made 2 pools, total of 88 amplicons, ordered primers, and the primers that we designed were based upon a first metagenomic sequence that was generated from a Danish patient, from one of our collaborators, Martin Schaub-Pedersen, from the Hospital in Denmark. And we also had some of the first sequences that were there from Portugal, went through a primal scheme and came up with the primers, ordered them, and tried them. And what we did, because we originated sort of from the SARS-CoV-2, we tried 2 basic approaches. So of course we did the standard method. So it was the exact same settings as in the midnight protocol for SARS-CoV-2 using the Q5 polymerase, which was about 3.5 hours than PCR to do. And separately on the next gen, we did it with a different polymerase, so the Arctic fog chemistry, to see if we can do it within 42 minutes. So that was sort of our setup, is to sort of copy it for the new pipeline. And in that way, compare 2 different PCRs. Now we changed slightly, of course, the bioinformatic analysis part. So the first is default, using the MinKnow high accuracy live base calling. In the analysis, we removed, of course, the short reads, the very long reads, used FastQC to see what the data quality was, to remove reads with average p-Phred value below 7, mapped it to the monkeypox virus genome from Michigan as a reference, which is also used in NextClay to look for specific mutations. Removed all the unmapped reads. That was basically to dehumanize the sample so we could share some data with other research groups, and used 2 different approaches to generate consensus sequences and also look at coverages. So via PySumStats and then generate consensus. And we use for SARS-CoV-2 also the True Consens package, which is from our national reference laboratory that designed this package. And it's also directly usable for monkeypox with some additional settings. And lately we also tried to tweak a little bit the consensus calling near homopolymer regions. So monkeypox has a lot of homopolymer regions where the R9 flow cells oftentimes call a gap when there's actually a sort of a nucleotide there, in comparison to many of the other reference genomes. So we designed also some custom Python scripts to actually do call nucleotides when they are detectable at a certain level in both forward and reverse read orientation. And there also has to be sort of a maximum number of mismatches at that position to be called the consensus nucleotide as well. So that worked really well. We had about 90 positions over the total genome, which are actually near homopolymer regions that give sort of a problem nucleotide calling, and it removed about 85% of 85 of the 90 positions due to the homopolymer regions. This is an overview about how the coverage is. So all of the separate amplicons. One of the first time in my entire career that the first experiment actually worked really well and amplified the entire genome. We noticed 2 amplicons that had slightly reduced coverage, so 22 and 50. We noticed this was primarily due to dimer formation with the primers, which we solved by doubling the concentration of the primers in both applicants to have a good coverage for both applicants. When we, sorry, we made a dilution series as well to compare the Q5 polymerase to the next-gen PCR. And what is obvious is that the Q5 works better at lower concentrations of the virus. So we see some dropouts occurring in particularly the low load samples for the next-gen, probably because of the very fast speed of the PCR that certain applicants that perform less well tend to drop out faster. And with the Q5, we still had good coverage and consensus for the entire genome. We also see about the percentage of N-calling. So with a CT value of above 25, we saw some very minor dropouts occurring. When we look at our clinical samples, the vast majority of blister fluid samples were actually below that value, which are very good for sequencing. So we could use most of our clinical samples in sequencing. Sorry, I have a bad throat. So we used this for one of the cases early on in the epidemic. We're one of the few countries that also had an infection in a small child, which we investigated quite intensively in Amsterdam, from epidemiological investigations and going to schools, going to sport clubs where the boy attended. And we were not able to identify the potential source of infection. So in the end, we tried it with sequencing, and this was one of the first sequences that we were able to generate. And we also looked at, of course, other random samples that we had from the sexual health clinic, from the MSM population. And we were not able to identify an identical genome from any of the patients near at the same time. So they were all very different. So that came to us that we had no really known source of infection. And one of the key questions also from the child itself and their parents was the stigmatization that they feared. So they were really keen on publishing this case to really show that there was no link, no sexual abuse possible with this child, and that any other forms of transmission were also possible with this virus. That could also be a non-sexual transmission case. And that really helped also in educating the people and the community that there are different ways of transmission other than just sexual transmission among men who have sex with men, to reduce stigmatization of infection of other people in the community. So that's where sequencing really helped at that time. A few of the key questions that were also like, is the sequencing reliable enough? This the assumption in the beginning was that this is a virus that is highly inflexible, invariable, and is there enough signal in it, or is the sequencing error rate not higher than the mutation rate that the virus will go through? These were sort of key questions to see how we can incorporate sequencing in the epidemiological investigations. So we went back to several sets of really epidemiologically linked pairs. And these are donor recipients are really couples that are known to have infected each other. And others are couples that went to either same source, of which they believed that was the actual same source, and had disease presentation or first day of symptoms around the same time. So this was epidemiologically the most clear cut that we could find. And within the 5 pairs, we see some interesting data. So in 3 of the couples, there were actually identical sequences being generated. We have a third where you see in the sort of the reddish dots, these were the pair that was sampled about 13 days apart, with only 1 or 2 mutations occurring among them. so they clearly clustered together within the tree as well. And the other one where a couple said we probably had the same source, actually visited a sex venue with a party going on, of which they had multiple contacts, also different contacts. So there it's questionable in the end whether or not it was the same source or a shared third party. We also looked at the other way around. So in a lot of the source and contact tracing, a lot of index cases mentioned the same location where they went, either at certain moments or not. And one of the questions was, is there sort of ongoing transmission at that site from somebody that we not sample? Are they different, or are they completely separate transmission events, but just at the same location being? And here from the individuals, we noticed there was actually 3 clear separate parts. So one to a location had no other linked cases, other ones of which went to the same party at the same night, and they had the same, nearly the same strains. And this was another case that went to the same party also at the same night as the other individual, but claimed, yeah, it may have come also from a separate individual who we had contact with, on which we clearly show that it's also done at the same location. So also that helped in trying to identify locations, or was it continuous separate events that were there, and also led to the fact that the same party location also served as an information night and a later moment where municipal health service workers and infectious disease doctors went to house an information event about monkeypox and the chance of monkeypox and identification of monkeypox. And after this, we had no more clear infections related to this location. Separately, we also retrospectively looked at how many cases prior to May 20th could we have missed. So we retrospectively tested within our sexual health clinic in Amsterdam all of the cases that had a positive STD, because when negative samples are not stored longer than 1 week within the laboratory, but all positive samples with another diagnosis are stored for a longer period of time. So we tested back to March 1st of this year and identified 3 additional cases that we previously did not know about, of which the first case was actually the 6th of May, so that predates the first known infection in the Netherlands by a few days, but no cases really prior to the beginning of May in Amsterdam. And we also see that at least a few, the first 2 also link or identical to many other of the very early samples that we had in the country or in the city as well. And the one from the 13th actually matched also with a lot of cases that became positive later on. So it looks like one of those early infections also seeded a lot of new infections later on in the outbreak in Amsterdam. So in conclusion, monkeypox virus can really be sequenced fairly straightforward with this amplicon-based approach. You can do the amplification using exactly the same polymerase and PCR protocol as for the midnight protocol for SARS-CoV-2 in about 3.5 hours. So you can also combine monkeypox sequencing with normal SARS surveillance that can be done. The 2 amplicons that generate heterodimers can be solved by just increasing the primer concentrations for these cases. And we see some amplicon dropouts when the load is very low, but this is mainly in the next-gen PCR, and it really hardly occurs in the Q5. So we can sequence up to CT28 and still get a full genome. And also the primers are available commercially from next-gen for this protocol, so they can be found online to order. Rapid design and the usage basically of tiled amplicon-based approaches combined with nanopore sequence for us in Amsterdam provided valuable insights in the outbreak and also helped guide public health responses. But it's very important to have a short turnaround time to help guide and also to have sort of good interaction with the public health community within the city to generate the questions needed to be asked, and also to have the proper answers to help guide them. And what we did observe is at least that the evolutionary rate appears to be higher than expected, making it useful more for genomic surveillance in comparison to what sort of we previously thought, that it was just 1 or 2 mutations for an entire year. So that was one of the revelations for us as well. And a lot of people to thank from the separate labs and municipal health service. And this if you want to come into contact, if people want to know like some practical experience or tips or tricks, do contact us. Myself, Marcel Jonges is our head molecular microbiologist at the Amsterdam UMC. Did a lot of PCRs and sequencing as well. And also from the Oudis from NextGen PCR, who knows all of the fast PCR machines. So I'm happy to take any questions if there are any.
Hi everyone, good afternoon. First, I would like to thank Oxford Nanopore Technology and the people involved in this webinar for the invitation and the introduction. My name is Ingra Claro. I'm based at Imperial College London as postdoctoral research associate, and the title of my talk today is Rapid Viral Metagenomics using smart RNA amplification and nanopore sequencing. Starting with a brief introduction, the RNA viruses are responsible for causing a broad range of human and veterinary disease. In the last case, they have been a cause of emerging and re-emerging infections, such as the viruses Zika, dengue, HIV, Ebola, and most recently SARS-CoV-2. All of these resulting epidemics and pandemics have caused a high morbidity and mortality and economic costs. To date, our ability to manage these outbreaks is hampered by the challenge of making definitive clinical diagnosis, as many of these viruses are often clinically undiscriminated from those caused by co-circulating viruses and also from some bacterial pathogens. And the diagnostic tests can also be limited by the low specificity in the case of serological tests, or can require a priori knowledge of the viruses to be targeted and are limited by the numbers of the viruses that can be tested at the same time. And that happens in the case of the RT-qPCR methods. In the routine, especially when you have an important epidemic happening, as we are seeing now, the tests normally are targets for viruses of public health importance at the moment. And for these reasons, acute febrile illness often remain undiagnosed, leading to a failure of epidemiological surveillance. That was previously demonstrated by Zika virus in Brazil. At the time we had the Zika circulating, we have another arboviruses happening at the same time. So the detection of the first case was delayed at least 1 year after from the real onset outbreak. So new applications have been driven by technological advances in sequencing. So here we have the first example of real-time genomic surveillance with the multiplex PCR approach that were conducted using target amplicon sequencing on the Minio sequencer. These studies allowed whole-genome sequencing directly from clinical samples, even in samples with low viral copies, and also exploited the portability of nanopore sequencing to archive a faster turnaround time by sequencing the samples close to where they were collected. Since then, several genome surveillance studies using target multiplex amplification have been carried out to characterize the different viral outbreaks. So here we have some examples, including the Zika virus in Brazil and the Americas, the yellow fever circulation in Brazil, and chikungunya virus, and also dengue 2 in Angola. And nowadays we have the SARS-CoV-2 that has now more than 1 million sequences submitted with the multiplex protocol. So while successful for the epidemics described before, this approach is best when the outbreak strain is known, but is less suited to diverse viral groups or for viral discovery. And for that, we have the viral metagenomics, that is the process of sequencing the total viral nucleic acid content in a sample. That can be the complementary cDNA or the DNA that allows the genome characterization of known and also novel viruses in an untargeted manner. So this technique is useful for diagnostic, clinical laboratories, and public health surveillance, and have already been shown promise for nanopore sequencing with the CISPA protocol, sequencing dengue and chikungunya, and also Lassa virus directly from clinical samples. Well, the limitation of our metagenomics approach when sequencing directly from clinical samples is that it can result in poor sensitivity, especially in samples with a low abundance of viral genome material relative to the host-derived nucleic acid. So we described two high sensitivity, low input, smart approach for nanopore metagenomics of RNA viruses. And here we validated from isolated samples and also from clinical samples. The smart board means switching mechanism at 5N of RNA transcript and was originally described in 2001 using oligo-DT priming to target polyadenylated mRNA molecules. So what it did was to adapt this method to use random priming for cDNA synthesis, followed by PCR amplification. So the SMART9N approach is based on the NAB next single cell from New England Biolabs. So we modified to use a random priming and native barcoding library preparation using the kits of Oxford Nanopore. So the New England Biolabs method uses single priming PCR amplification, which we found we could perform using barcoded primers for the rapid PCR barcoding kit available from Oxford Nanopore. So we decided to modify the sequences of the RT and the SSP primers for the rapid SMART9N protocol that enable the addition of the barcodes in a single step during the PCR reaction. So the methods were validated in Zika virus isolate samples and in yellow fever and SARS-CoV-2 directly from plasma and residual nasopharyngeal samples. So the clinical samples had CT up to 34, and the SMART9N and rapid SMART9N protocols were compared to the gold standard multiplex PCR method. So here we have the comparison between the complexity of the methods and also the time and costs. And compared to the multiplex PCR, the total time of hands-on laboratory work dropped 15% and 61% for the SMART9N and rapid SMART9N respectively. And reagent costs were reduced by 40% and 45%. For bioinformatics analysis, we used a quick and easy to run bioinformatics pipeline consisting of basic calling, using base caller, primary timing, and the multiplexing using PartChop, alignment of each FASTQ file to the genome reference using MinMap2, and then followed by some tool sort to convert the FASTQ files to the BAN file. So Nanostat and tablet software were used to calculate the genome statistic, as well as the Santus stats and Santus Deep. And for the multiplex PCR assay, the statistic analysis and consensus sequences building was performed using ARTIC bioinformatics pipelines with the nanopolished version. For the SMART9N and rapid SMART9N assays, variant calling was performed using Medaka variants, and the consensus sequencing was constructed using margin Medaka consensus, both from Oxford Nanopore. Finally, to search for other viral RNA in clinical samples, we did the taxonomic classification using Kraken2 and the database of the mini Kraken2 8 gigabytes. The initial test was performed using Zika virus control sample, which was subjected to SMART-9 assay at the dilutions up to 1 to 10,000. The test resulted in nearly 100% genome coverage for all the dilutions tested and with a coverage of up to 3,000. And for the dilution 1 to 10,000 in particular, we got the whole Zika virus genome in one single read with an input of only 6 picograms, that is an input compatible with already available single-cell assays. After that, we used the same sample to compare the three methods and also to analyze the limits of the detection of the metagenomic methods. So as we can see here on the coverage plot, the metagenomics protocol had better genome coverage across the genome when compared to the multiplex PCR assay and higher deep coverage across the genome. From the 0-10-fold dilutions, we analyzed dilutions up to 10 raised 1, and mapped viral reads were detected from all the dilutions tested for the two metagenomics workflows. So the minimum genome coverage was 90% for the SMART-9 assay and 42% for this rapid SMART-9 assay. We next apply the validated assays to representative yellow fever clinical samples. So here we can see the coverage pattern of the three methods from the mean depth of coverage at a 95% confidence interval. And as we saw earlier, we could observe a better coverage pattern through the genome for the metagenomic methods when compared to the gene-specific one. And also we obtain mapped reads in all the assays and in samples with CTs up to 34. The same was observed for SARS-CoV-2 clinical samples sequenced by the rapid SMART-9 approach. We could detect reads mapping to the reference virus genome presented in all samples with genome coverage of 100% in all samples tested. And here you can also have a concordant coverage dip and pattern across the genome, and which is very important to track the virus divergence. We also compared the results of the N50 value of the approach, and we got an N50 of around 2,000 base pairs for the yellow fever samples and up to 2.6K base pairs for the SARS-CoV-2 samples. And here, when analyzing the read individually, we observed approximately 60% of the SARS-CoV-2 genome in a single 18.5K base pairs read. And as far as we know, is the longest published viral cDNA read to date, produced by our rapid SMART-9 method. Using this approach, we could identify the Sabiá arenavirus in 2 fatal hemorrhagic fever cases in São Paulo, Brazil. These cases were from suspected yellow fever patients. And the Sabiá virus is a pathogen responsible for causing hemorrhagic fever, described in the 90s in Brazil. For the last 20 years, only 4 human Sabiá virus infections were reported, being 2 of these infections naturally occurred in different regions of the São Paulo state. And the other 2 infections came from laboratory environment infection during the identification of the virus. In January 2020, we had 1 case of Sabiá virus reported to the surveillance center in São Paulo. And We then performed a more extensive characterization of the virus using the Smart-RNA metagenomic sequencing of the samples. And we also decided to investigate the samples of patients with suspected yellow fever virus infections with hemorrhagic fever between 2018 and 2019. That was the years where we had a higher number of yellow fever cases in Brazil. During the investigation, we have detected Sabiá genome fragments in one more fatal case from 2019, that is the case 3 reported here, and this case had a very low viral load. So we put together the sequences of the 3 cases, 1, 3, and 4, because the case 2 we didn't have the sequence available, and then we performed a phylogenetic analysis of this case, and we could show different strains from the case 3 and the case 4. Also, when analyzing the genetically diverse of this case, we saw that this case had a higher than 10% nucleotide distance at the nucleoprotein gene when compared to the other, to each other, and also when compared to the reference virus sequenced for the 19th. And because of that, we decided to analyze the currently molecular tests to detect Sabiá virus infection. And while analyzing the sequences generated by the Metagenomics protocol, we found out that the currently PCR tools would not be able to detect the circulating Sabiá virus strains due to the genetic diversity of the viruses at the primary biogen sites. So we then designed a new in-house PCR and nested PCR detection protocols to test the Sabiá possible circulating strains, and with high and also with low viral titles. So here at the first figure, we have the RT-PCR with 6 primer sets that we designed, and figure 2, we have the nested PCR for the low viral samples. We used the case 3 and the case 4 to do the standardized of these tests, and then we validated these tests with the positive samples. And then we used these tests to test all the healthcare workers who had contact with the most recently patient presenting Sabiá virus infection in the Hospital das Clínicas in São Paulo, and who revealed no secondary cases. So the structure described here from the metagenomic identification, the development and validation of the molecular methods, and also the testing of the all healthcare workers was conducted in a total of 10 days, proving to be an enhanced pathogen detection technology for diagnosis in real time and for surveillance. Now, switching to another virus, our group described the first case of monkeypox virus in Brazil. As was explained very well in the last talk, in early May 2022, cases of monkeypox virus were detected in the UK and Portugal, most of them with no travel history of endemic countries. As of June 9, we had more than 1,000 confirmed cases on all continents, most of them in the European countries and in the United States. In South America, the first 2 monkeypox cases were reported from Argentina on May 27, and they made available 2 partial genomes of monkeypox virus as the first sequences from Latin America. As of June 8, Brazil had already 8 suspected monkeypox infections and in 6 different states, and we could confirm the first case in São Paulo on June 7 from a patient with a 41-year-old male with a recent travel history to Portugal and Spain. From this case, we received a skin swab of the lesions, the vesicle and the crust, and then the samples was sent to us, and we use a shotgun metagenomics workflow, also using the rapid PCR barcoding kit from the Oxford Nanopore. So the protocol consisted of the DNA extraction from the swab samples, and so here we can do our RNA depletion depending on how you are extracting the material, and followed by DNA fragmentation using the fragmentation buffer of the kit, and a haptic amplification with the barcodes was performed, followed by cleanup, quantification, and quantification of the PCR products. The MinION libraries were prepared by adding the adapter via click chemistry, and the final libraries were loaded in a new R9 flow cell on the MinION device. And here we continue using carry out using Mino software with the default 48 hours run script. So we didn't left for 48 hours. We left the run overnight for around 12 hours, and we got almost 1 million reads with almost 800 reads with quality score higher than 8, and then this reads was used for the following analysis. And here we used the same pipeline described for bioinformatics, and the average zip was almost 300, covering 100% of the viral genome with at least one read and within 50 of 4,000. So to contextualize the novel monkeypox genome, we downloaded 102 whole genome sequences from the Sabiá, being H1 from the ongoing B1 multi-country outbreak. As expected, the first case of monkeypox virus in Brazil clustered within the newly proposed B1 lineage and was closely related to sequences from Portugal, Germany, USA, and Spain. And here from this sequence, we identify 3 unique SNPs when compared to the genome reference, and we then investigated the mismatches in the binding sites of primers and probes of the available molecular tests. And analyzing that, we found 2 mutations in the binding regions, but according to the position of the mismatch, we saw that it would not affect the sensitivity of the primer sets, indicating that the existing primers are capable of detection infections from the current outbreak. 1 week after, we received a second suspected case from a 27-year-old patient with a recent travel history to New York. And this patient was from Manaus, Brazil, but had been living in New York for the past 6 months. So he reported that the symptoms started on June 3rd, and on June 9, the skin rash started to appear, first on the genitals, followed by the hands and the belly. By June 11, he returned to Brazil, and after 2 days, he was hospitalized, and the samples collection was done on June 14. In this case, was collected the whole blood, urine, nasal, and oropharyngeal swabs, and the skin lesions were collected, followed by the shotgun metagenomics sequencing. On the same day, we got the results, the positive result of this case, and the sequences were submitted in around 10 hours. And it was possible in this case because we used the Humpert for real-time analysis. So with 1 minute of sequencing, we could get the results having 66 reads mapped to the monkeypox virus genome, covering about 70% of the virus genome. So after 1 hour, we reported 99% of the monkeypox genome with an average zip coverage of up to 10. And for the final analysis, we had 1.2 million reads in total. And as I said before, here we we included all the samples in the sequencing, but we found reads mapped only to the lesions samples, with genome consensus covering 100% of the genome for both, with a deep coverage of up to 150-fold. So after that, we saw that we could start to multiplex more samples at the same run, not only one sample in the same reaction, using Rampart for real-time analysis, which decreased the cost of the method. Concluding, a low-cost, sensitive viral metagenomic surveillance systems are needed to rapidly identify novel and divergent viruses. This can be critical for public health, as the shortage of data of infection agents represents an obstacle to early detection, prevention, and containment strategies of potential epidemics. So here we demonstrated 3 sensitive metagenomics workflows across viral, isolate, and clinical samples, which take advantage of long-range nanopore sequencing technology by generating long cDNA amplification products for viral metagenomics. And with the rapid SmartNN and shotgun metagenomics, we could improve the cost and complexity of the laboratory work, promising for routine use and in research laboratory as well, and as well in the field. If you are interested in any of these protocols, please check the studies here. We have the detailed protocol published and also the studies of the metagenomics and the monkeypox cases. I would like to thank my group in Brazil from the Institute of Tropical Medicine, and especially Professor Esther Sabino, that was my supervisor during my PhD. The Nick Lohmann's lab group from the University of Birmingham, where the methods were developed. In particular, Dr. Joshua Quick, that made everything possible for this project to happen. And my group here in the UK at the Imperial College London, and in particular my supervisor, Professor Nuno Faria, for all the support. So here is our contacts. Please send an email if you have any questions, and I